AI-discovered medicine is no longer just a prediction about the future. Researchers have already used AI to identify a possible disease target, design a new molecule, and move that molecule into human trials. In one randomized study, patients on the highest dose showed an increase in average lung capacity, while the placebo group got worse. Now, a much larger Phase III study has been registered. This isn't an approved treatment, and it isn't proof of a cure, but it is concrete evidence that AI can help scientists go from biological data to a real experimental medicine and to measurable results in real patients. So what exactly did AI contribute here? And could this approach make new medicines faster? Here's the clearest way to say it. AI helped point to a target inside the body that hadn't been tested before. AI helped design a brand new molecule aimed at that target. That molecule moved through lab testing, early human safety trials, and then a mid-stage trial in real patients with a real disease. That's a genuine, verifiable achievement. At the same time, this medicine is still experimental. No regulator anywhere has approved it. A promising signal in a mid-stage trial is not the same as proof a drug works. Both of those things are true at once, and this video is going to hold on to both. The disease at the center of this story is idiopathic pulmonary fibrosis, or IPF. In IPF, lung tissue slowly turns stiff and scarred, the way fabric hardens over time. As scarring builds, the lungs can't expand the way they used to, and breathing gets harder. There's no cure. Approved IPF medicines can slow the scarring down, but they don't reverse it. Most people are diagnosed later in life, and typical survival after diagnosis is only a few years. That's the real stakes here. A treatment that could genuinely slow this disease down would matter enormously. So where does AI actually come in? The first job was picking a target, a specific protein a drug could act on to fight fibrosis. Researchers at Insilico Medicine used an AI system trained on huge amounts of biological data, gene activity, protein networks, research literature, to rank possible targets for IPF. One protein rose to the top, a kinase called TNIK, sitting inside pathways linked to scarring and inflammation, and at the time, a largely untested idea for this disease. It's worth being precise here. The AI didn't decide anything on its own. It surfaced a ranked, data-driven hypothesis. Scientists still had to review that evidence, design experiments, and confirm in the lab that TNIK was worth pursuing. Once TNIK was chosen, the harder problem was designing a molecule that could switch it off safely inside a living person. This is where generative chemistry came in. Think of TNIK as a complicated lock and a working drug as a key shaped precisely enough to turn it. Instead of a chemist hand-drawing candidates one at a time, an AI system generated thousands of possible molecular structures, then predicted which ones would bind well, behave safely, and could actually be manufactured. That narrowed a huge search down to a small handful of real candidates worth synthesizing. And that distinction matters. A promising structure on a screen isn't proof it works. Every candidate still had to be built and tested the old-fashioned way, in real chemistry and real biology. That candidate eventually became Rentosertib. After clearing safety testing, it reached a mid-stage human trial called Phase IIa. 71 people with IPF, randomized, double-blind, and placebo-controlled, meaning neither patients nor doctors knew who got the real drug. People were split into 4 groups for 12 weeks. Placebo, a low once-daily dose, that same low dose twice a day, and a higher, 60 mg once-daily dose. The key measurement was forced vital capacity, or FVC, essentially how much air someone can forcefully breathe out after a deep breath, a standard way doctors track IPF. After 12 weeks, the placebo group's lung capacity got worse, dropping by just over 20 milliliters on average. The highest dose group moved the opposite direction, improving by about 98 milliliters. That gap, one group worsening, one group improving, is exactly the kind of signal that makes researchers want to run a bigger trial. Here's the honest caveat, and it matters just as much as the result. Each group had fewer than 20 people, small enough that ordinary bad or good luck can swing the numbers, and the statistical confidence ranges here were genuinely wide. The trial ran just 12 weeks, not long for a slow-moving disease like IPF. Not everyone finished it. Some participants stopped early because of side effects, including some cases involving liver-related lab values. None of that erases the finding, but it does mean this was a promising early signal, not proof that Rentosertib actually helps patients over time. That difference between a promising signal and a confirmed benefit is exactly why a bigger trial exists. That bigger trial is Phase III, designed to answer what Phase IIa couldn't. It's planned to enroll around 320 patients, again randomized, double-blind, placebo-controlled, but following people for a full 52 weeks instead of 12. Its main measurement is the yearly rate of lung capacity decline, a much stronger test of whether Rentosertib actually slows the disease, not just moves a number short-term. As of late July 2026, the trial is registered but listed as not yet recruiting, with an estimated start date of August 30th. In other words, designed and registered, but patients haven't started receiving the drug in this next phase yet. Zoom out, and Rentosertib is really proof of a bigger idea. The same kind of AI system could, in principle, search huge biological datasets for other disease targets, generate brand new molecules for those targets, predict which ones are worth testing, and help rule out weak candidates before years and millions of dollars get spent in the lab. The FDA has said it's seeing a real, ongoing rise in drug applications that include AI components across development. That's a meaningful trend. It does not mean hundreds of AI-designed medicines are already on pharmacy shelves. It means AI is increasingly part of how new medicines get built, one careful step at a time. None of this replaces scientists, and it's worth being direct about why. AI proposes a target or a molecule. Real experiments test whether that proposal holds up. Scientists interpret what those experiments mean. And clinical trials, run and overseen by human researchers and regulators, ultimately decide whether a medicine is safe and effective enough for real patients. AI is becoming a genuine scientific partner in that process. But every decision that matters for a patient's safety still runs through human hands. So, back to the question we started with. AI has already shown it can meaningfully help identify a drug target, design a real molecule, and push that molecule into human testing. Rentosertib's Phase IIa result was promising enough to justify a much larger study, but Phase III is what will actually determine whether that benefit is durable, clinically meaningful, and safe over a full year. The question is no longer whether AI can help design a real experimental medicine.